And now we go to one of the highlights of the meeting today. We always like to finish strong, and we're finishing really strong this time. This is one of the companies that got the most interest since I work at Optica, and it's been almost a year so far. Halitus, medical doctor Sven Youngman, and CTO Joanna Pavia, thank you very much for letting us go the way that we deserve with one of the highlights of the meeting. Halitus, the floor is yours.
Thank you so much, and thanks everybody. This has been a joy to watch. I'm here from Berlin. I'm a medical doctor originally, so don't expect me to go too much into the technical details. But I'm speaking to you from the user perspective on the end of the day, which is important for us in our development, that we closely intertwine software development, hardware development, and then the clinical perspective. I have with me Joanna, as Jose said, thank you for that, who, as you will see, is a fabulous engineer as well, and is mostly in charge of everything around the data perspective, but she also has a PhD in photonics. So she can take care of the additional questions. This is what we're building. I'll go into it in a moment, but the bigger story here is, we have taught machines how to see. We have taught machines how to hear. The next frontier in machine sensing, on our view, is smell. And there's a lot to be gained from that, right? The thing that we focus most on is the analytics of dynamic gases versus, your smoke detector or something. And within that, and that's also why I'm part of this whole journey, we want to look into the breath, to decode the breath. The larger big insight here that people have really been amazed by over the last maybe decade or even more is that every breath contains more than 1,000 molecules, 1,000 different molecules, I should say, right? And many of these molecules are volatile organic compounds, which reflect things that are happening in your bodies, your metabolism, and so on. And what's exciting about this is that it gives us a non-invasive, real-time, more or less, insight into a lot of things. You know that rats can smell diseases, or dogs and cats can, and so can machines. So a lot of the research that has been built on this and where we know these things from, they mostly come from the technologies around gas chromatography, mass spectrometry, and so on. And there are some other devices, you might have heard of e-noses, but the way we see it is they're all very limited when it comes to scalable clinical applications, because many of these devices are very sensitive. They need a lot of specialists to operate them. You have consumables that you need to use on end, and quite often they end up being very in sort of a centralized diagnostic setup, whereby sampling has to happen somewhere else. And by the way, sampling is one of the biggest concerns, because how do you know that what you've been measuring really comes from the breath? And how can you differentiate if it comes from the upper airway, the lower, or somewhere in between? Which does make a difference, because some of these biomarkers are produced in different parts of your lung. And what we've been doing is we developed essentially a photonics component and combined it with a gas dynamics component. So what you see here, the center, which basically looks like a Dyson hairdryer, if you will, this is a spirometry. So the thing you know from your lung doctor, nothing special. It detects the movements, the gases, and so on. And to that, we combine what we call the sort of the optical chambers, where this is just 6 meters in diameter, but it can reflect the laser up to 8 meters. So you have a wall of laser essentially, and we put several lasers sequentially that are each triggered on very specific biomarkers, of which we know that we need to find them. So this is a tunable diode laser absorption spectroscopy. And in effect, what it allows us to do is we can know what the air looks like before somebody has been blowing into this. So we have a baseline assessment. And then we can do 10 cuts per second, which enables us to do a full end-to-end breath sequencing with data on the exact gas streams and with data on the concentrations of certain biomarkers of interest. And the exciting thing about this is, and I can say that as a doctor, and we hate to say that quite often, is this will be able to be put in the hands of medical lay people. You don't need to have a medical degree. You don't need to be a nurse or lab technician. You can be a security person at an airport. And the second cool thing is that we attempt to make this result show up in 1 minute. So if you compare that with, for example, a PCR for COVID, which in the express version, which is very expensive, is going to provide you with a result in 30 minutes, and somebody needs to swab you in the nose, and they need to send it somewhere. And we can say we do it in a minute, and we could even, send what we have here, like a geotagged signal onto a map, so that authorities or aid organizations see in real time where the hotspots are in an outbreak. And this doesn't have to be COVID primarily, right? We can also talk about tuberculosis, where still in Africa, 44% of the people with TB are not diagnosed, and they keep infecting others. Or in the US, 20% of immigrants from certain regions have TB, right? So doing a quick screening there is quite interesting. Same for schools. How do you make sure that children with infections don't come in and infect others and then infect their parents and so on, with a rapid screening, right? You can't do that with a 30-minute delay screening, or not even with a 15 minutes so easily, not on a scalable basis. So that's it, right? This is photonics, we think is the way to go. It's the technology that is underestimated in the space. People look at all sorts of chips, they look at mass spec and so on. But this is the only thing that really allows you to do an inline sampling, if you can even call it a sampling, because it's not really, that is easy to use, portable, and provides fast results. If you want to do a Q&A, so then...
No, we could continue. Joanna, take it over from here.
Yeah, so probably I will continue what I would think that it's a challenge, or some challenge for us, and also for the photonics community. So because as a background, as Sven presented, so I'm a biomedical engineer, but with a PhD in physics. So I've been working for the past 10 years in photonics technologies, but also with signal processing, machine learning, mostly for disease prediction and so treatment personalization, and also for predicting diseases in early stage, and doing personalized medicine. So there is a challenge that we are facing now, and I've faced so in the past. So because I was working also with optical fiber sensing, so trying to measure biomarkers in fluids like blood, using guided optics for diagnosing diseases like Alzheimer, for example. And now we are I'm building this fabulous technology that is the creation of new digital biomarkers. So basically, you have, of course, the validated biomarkers like the proteins associated with these diseases. For example, in Alzheimer, you have the beta 42 for that is a protein specific that you can found in cerebrospinal fluid, and you have like optical fiber sensors to do that. So in this case, in breath analysis, you have volatile organic compounds, small molecule concentrations, and also lung function parameters. But the question is that there is a lot of studies and literature, so of course effective, but not effective enough in order to predict some diseases. So, and I believe, and I think that the scientific community believes that there is space for new biomarkers based on, for example, the derivation of these biomarkers. So, for example, signals related with the physics of particles, signals related with movement of for example, how people blow the air into the device in the case of breath analysis. So the frequency, how they do that. So other physics-related biomarkers that are not well validated yet. And so my question and my challenge is, at which level and when it's exactly enough so in terms of validation for the scientific community and also for the medical community, accept these new biomarkers as digital biomarkers also for using for screening in medical care and also for clinical usage. So
thank you very much, Joanna. And I can tell you that the reason why I'm so excited about this is because in my simple world, you are diagnosing lung cancer by simply measuring the breath exhaust with a handheld device. This is what you are doing. And this is so, so impressive. How can we help you? Because we really want to help you. I mean, I don't want to organize more telecom meetings at OFC. I want to help you. How can we help you?
I mean,
yeah, exactly. So please
no,
go
you
on,
go.
Yeah, so this question about so this new digital biomarker scene, the explainability of the signals that we are getting, so of course that are based on science, but so they are new. So we need a new community based on with background on AI, but also on photonics in order to help us to validate and convince the medical community that we have real information here. And secondly, so we are also building a big IoT infrastructure with centralized information, and we want that so this handheld and portable device for example, 100 device could be able to deliver information to our cloud system and process and to do predictions for detecting lung cancer in 1 or 2 minutes. So this is a big...
Dr. Levin, when you see this, I mean, I don't know if anybody in the room is getting the goosebumps that I get when I hear this. What do you think the Massachusetts Medical Center could do to provide some help, some assistance of us having this device as soon as possible in our hands?
Well, that's a very good question. First of all, I think it is amazing and really wonderful to be at the point where we're going to detect these diseases, and especially lung cancer, with a simple point of care diagnostic. I think that's amazing. In terms of Harvard Medical School and Massachusetts General Hospital, certainly there would be great interest in implementation of such a technology. I don't know if you were asking specifically about, clinical trials or
Yes.
ways to bring it to the public. So we do have a clinical trials unit specialized in doing this. And I am not even though I do clinical studies at the research center and I do it on people, of course, that's related to skin, but I could certainly connect you with people if you were interested in exploring doing a clinical trial at MGH. Of course, you have many potential outstanding medical centers at which to do it, but yeah, congratulations to you, amazing, and I'm happy to help move things along.
One of the biggest consultants in our network is in the room. Richard Crocombe, consultant for spectroscopy solutions, is in the room. Richard, I don't know if you heard this and you get the goosebumps that I'm getting. This is a breath analyzer that can diagnose potentially lung cancer. How can we help? How do you think we can help?
A more general answer. I mean, I'm generally very excited by all the miniaturization of optical technologies and what that means for wearables, incorporation into consumer devices, all kinds of different areas. And I think that the things we talked about today in terms of silicon photonics, photonic integrated circuits, etc., have enormous promise in this area to bring the cost down, make things portable, make things accessible. And so I'm just
Richard,
generally very excited about the whole area.
Quantum cascade laser. So it is a quantum cascade laser. It's expensive. Yeah. It's expensive, but this guy needs cheaper quantum cascade lasers.
Oh, absolutely. I mean, historically, sort of one QCL chip was $10,000, from well-known suppliers in the area. And so bringing the cost of those down, and there are a number of people working on QCL arrays for various applications, including safety and security type applications. So QCL arrays, I think, the potential for low-cost QCL arrays, I think, are huge.
We also have in the room Shakiba Kabe from Oxford Instruments. Shakiba, what do you have in mind? So I'm going to read her question. Yes, Shakiba, tell us.
Yeah, I've read the question, so I can answer. Yes, exactly. So Shakiba was asking me if we have to take in account the variability among the type of patients that we are including in our AI models that we are creating. For example, with our technology, we are creating models, for example, for detecting lung cancer, but for example, for the detecting and predicting exacerbation in COPD patients after they happening. So that is very valuable for physicians. So yes, we are considering cohorts with different parameters in terms of clinical comorbidities, so gender, sex, in order to test the models that we trained in an independent in a completely independent test, and then we can prove that our algorithms could be robust in different cohorts all over the world.